Today we will continue our discussion about this allogenic NK cells immunogenic effect by these host T cells. So in this figure we will understand whether this exogenous expression of this PD-L1 on this allogenic NK cells will have any effect on this host T cell response or not. So the experimental overview is shown in this panel A, where what it is, they take the allogenic NK cells and then transduce them with this PD-L1 lentivirus, and so that this NK cells, allogenic NK cells, will have this PD-L1 lentivirus, and then co-culture them with host T cells at different ratio. First, to check the activations of these T cells, they co-culture them at 1:1 ratios for 12 hours, and then check this activations of these T cells through the T cells markers, that is CD69 and CD. This is the experimental overview. In the panel B, it shows the lentivirus transduction efficiencies, where we can see that around 56.9% of the cells are transduced, which has this PD-L1 positive signals. Thereafter, they sort these cells and expanded these cells, and then use those cells for the final co-culture with the T cells. And in this right side of this panel B shows the histograms of this PD-L1 positive cells. That can be seen that it is higher than the background signals. So after co-cultures, they in the panel C, they shows the results for the CD69 markers, which is one of the markers for the activation of the T cells. Here we can see that when they co-culture them with this autologous NK cells, that is NK cells from the host cells itself, they do not see any activations of the T cells. However, when they co-culture them with this allogenic NK cells, when this PD-L1 is not expressed, they see high expressions of the CD69 markers, around 36 to 45%. But this percentage goes down to around 25% when this PD-L1 is been expressed. And similar results they have seen for the CD25 markers. Here also for the autologous NK cells, there is no activation signals. It is comparable to the background. And whereas for the case of this allogenic NK cells without PD-1 expressions, the activations, the CD25 markers percentage is high, around 15%, but it goes down to 7% when they used PD-L1 expressed cells. And when it comes to the proliferations of the T cells, they are able to see the high proliferations. It's around 15% in the case of the allogenic NK cells without PD-1 expressions, but it goes down to same around 7% in the case when they use PD-1 expressed allogenic NK cells. Thereafter, once validating the activations of these T cells, and they also see some suppressions, the partial suppressions of the T cell response against the allogenic NK cells with the expressions of the PD-L1, they check the cytotoxicity effect of the T cells at higher effector to target ratios. That is around effector to target ratios of 10 and effector to target ratio of 20. Effector cells in this case are the T cells, and the target cells are the allogenic NK cells. So for the control case, that is when they don't have this exogenous PD-L1 expressed on the allogenic NK cells, they are able to see high lysis, and this lysis of these NK cells increased in a dose dependent manner. That is, with high effector target ratio, they see high lysing of these NK cells, and this NK cells lysis percentage goes down when they use this PD-L1 expressed allogenic NK cells. So this further substantiates their previous finding from the activation markers that the PD-L1 expression of these exogenous allogenic NK cells suppress the immunogenic response or partially reduce the host T cells response. So this is what they have concluded in this figure. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.